One moment. Oh, no. Just a little. Oh, geez. Okay. Okay, can I start?
Yes.
Okay.
Yeah.
So it's a big pleasure to meet you to present Dr. Christopher Sanders from the Pirbright Institute. Thank you very much for accepting the invitation for giving a talk. I will talk briefly about your credentials. So Dr. Sanders works in the Pirbright Institute, beautiful place in the United Kingdom, and he got a degree in biology and oceanography in Southampton. I don't know if I'm speaking right, sorry for that. And then a master's in Imperial College in applied entomology. After that, a PhD in the University of Southampton. And since 2006, he works in the entomology group in Kilocortis in the Pirbright Institute. I have met Chris some years ago in a network project that was very challenging, and it was a big pleasure to work to know Chris and work with him in the last years. So I'm pretty uh, impressed with the excellent work and installations that he has in Pirbright. So it will be a great pleasure to see your presentation. It's I'm quite happy to see you again and very curious to see the novelties in the Kilocortis field. Thank you, Chris. So it's up to you now.
Thank you, Fernando. Thank you for the invite to speak to you today. It's a pleasure. Sorry I'm not with you, but those are the times that we live in. So as Fernando says, I'm going to be talking about Kilocortis biting midges and their role as vectors of Bluetongue virus. Quick introduction to the Pirbright Institute. The Institute's a world-leading centre of excellence in the research and surveillance of viral diseases of farm animals, so livestock, and viruses that spread from animals to humans, so some zoonotic viruses. We have both sort of fundamental research, so what we call blue sky research, which we just want to know what's going on, and also kind of applied research for answering problems associated with disease in livestock systems. And we have reference laboratories for some of these viruses as well. The viruses we work with are the reason for our existence is to work with high consequence exotic viruses, so viruses that don't occur in the UK. And perhaps the biggest of this and the reason why we have our facilities is that we can work with foot and mouth disease virus, which is a highly contagious virus of cloven-hoofed animals, exotic to the UK and has huge trade implications if you have the case. So we also work with Bluetongue virus, Pestipati ruminant virus, lumpy skin disease virus, African horse sickness virus, African swine fever virus, Rift Valley fever virus, dengue virus, chikungunya virus, and some chickenpox viruses as well. So we have a very much a viral theme, and largely our work is on the disease of livestock, but we do have some of these human pathogens as well. And just to let you know, we're in the south of England, about 40 minutes on the train from London. So in this talk, I'm going to introduce Kilocortis biting midges. You can see an example of that on the slide. Talk about Bluetongue virus itself and how the epidemiology of Bluetongue virus has changed quite dramatically in the last 25 years or so. I'll introduce some of the work we've been doing at Pirbright on Kilocortis and Bluetongue. And this will involve the work I've done on Kilocortis dispersal, impact of climate change on Kilocortis and Bluetongue, and then look at some virus vector host interactions that we've been able to study in the entomology group. So this is an overview of the work I've been involved with Kilocortis over the last 12 years, nearly 13. Okay, so here are Kilocortis biting midges. These have a near worldwide distribution. The only places that don't have Kilocortis are the Hawaiian Islands and New Zealand. Of course, we keep going back to look in Hawaii to make sure, but so far no Kilocortis in Hawaii. There's about 1,500 species of Kilocortis worldwide and about 50 in the UK. They are tiny. They are some of the smallest hematophagous flies. They're 2 to 4 millimeters long. You can see on my slide, you've got the sort of tsetse fly, which is a beast that's up to 17 millimeters in length. The Aedes mosquito, you'll be familiar with the size of that. And then Kilocortis are much, much smaller, slightly smaller than sandflies even. This makes them really difficult to work with, but we'll get onto that later. Kilocortis have these lovely patterned wings. I hope you can see on the right-hand side of the slide. And this helps us to identify them to species or group level, although some species are morphologically indistinguishable. And we do have some cryptic species as well. So we can use molecular techniques to identify those. One of the typical things about Kilocortis is their huge local abundance. We can collect hundreds of thousands of these insects very rapidly. My personal record in quite a small trap was over 65,000 individuals in a trap that was running, a light trap that was running for only 12 hours. The world record, as far as I know, is 1.5 million insects in 24 hours in a standard light trap. So there's a huge amount of biomass of these insects. Along with that comes a huge biting rate. So Kilocortis biting midge females need either obligate blood feeders or some of them are autogenous, but most are anautogenous. And they require a blood meal for the maturation of their eggs. And you can get huge biting rates of these insects. So the Highland biting midge in Scotland is particularly voracious, and you can get hundreds of bites per minute on just your forearm. It's a real biting nuisance. Because they're so small, there's a really strong influence of meteorological conditions and climate on their behavior, on their presence and abundance and activity. In distinction to mosquitoes, Kilocortis breed in wet organic matter, so not standing water or containers like you'd expect with mosquitoes. They need animal dung, leaf litter, fungi, enriched soil for their larvae to live. And hopefully, yes, there we go. So this is a video we had made a few years ago. This is Kilocortis life cycle. So here's the eggs. They darken as they age, and they hatch into these vermiform larvae that go around eating bacteria and protista in their environment. Normally they're walking through a kind of more of a substrate than this. And then they pupate, and after about a week to 2 weeks, again, temperature dependent, they emerge into these adults, the female emerging there, and then they blood feed. You can see this fully engorged female sucking my colleague Jim's thumb. Very difficult to get those shots. We had to have a specialist photographer for that. So when we think about Kilocortis and animals, we're thinking about farm animals in particular. We pretty much provide everything they need on a farm. They breed in animal dung, in soil and manure, so dung heaps, things like that, and where animals have churned up mud with water and manure as well. So we can find them on every farm. They're pretty much ubiquitous in the UK, and it's only in extremes of temperature that you can't find them. So associated with their role, their blood feeding habits of the females is this potential to act as biological vectors of viruses, largely of livestock. So I mentioned some of these in my beginning slide. So we've got Bluetongue virus that's transmitted by Kilocortis, African horse sickness virus, epizootic hemorrhagic disease virus, Schmallenberg virus, and the only human virus that's transmitted by Kilocortis is Overwintering virus from Central and South America. These are all, Bluetongue and African horse sickness are really quite nasty diseases. You can see on the on the right-hand side, a sheep infected with bluetongue, and a horse that's actually died of African horse sickness virus. It's a really nasty, highly lethal virus. And then EHDV is a disease of ruminants, but also particularly affects deer and is a severe problem in deer farms in America. Relatively few species out of the 1,500 or so are actually implicated as vectors. And it seems to be that we have groups or species or groups that are associated with transmission in different places. But this is quite flexible, and part of it is because we haven't actually studied a lot of these systems fully and haven't had the sufficient taxonomic expertise to actually identify them. So for example, we got Brevitatis and actini in Australasia. Kilocortis amicula is a European, and we call it African and Asian species. The Obsoletus group is in Europe and North America. And then we have Pararensis in Central and South America that's associated with the transmission of Arthropod virus. And then Insignis and Kilocortis sonorensis that are in the Americas. So each of these plays at least a role, if not the dominant role, in transmission in these different regions. But as you can see, they're actually split across different subgenera of Kilocortis. They're actually quite different. So what is Bluetongue virus? Bluetongue virus is the virus I will talk about. We do work on some of the other viruses as well, including African horse sickness and Schmallenberg virus. But we've been working on Bluetongue virus for a longer time, and it's a little bit more interesting until perhaps recently. So Bluetongue virus is a non-contagious vector-borne virus. It's an agent of bluetongue disease in ruminants. Bluetongue is a notifiable disease. So if you have a case of bluetongue, you have to notify the OIE, which is like the WHO for animal disease. It's exotic to Europe. So we don't expect there to be endemic virus in Europe. It's spread solely by the bites of Kilocortis biting midges. We don't get contact transmission, except, there's always exceptions to a good rule, for a couple of the more recently identified bluetongue serotypes, which do appear to be transmitted by contact and are not transmitted by Kilocortis biting midges. They're very similar, but they're not quite bluetongue. But they're called bluetongue. It's up to the virologist to decide where they actually fit. We usually only see severe disease in certain breeds of sheep, particularly those that have been improved by Western and European breeds of sheep, and in some species of deer. Cattle usually act as asymptomatic reservoirs, as do a number of deer species as well. So they're not going to show any clinical signs, but they may well act as a reservoir and act as the source of virus that then goes on to infect sheep, where we see the damage. The virus causes injury to the vascular lymphatic epithelium inside affected sheep, which results in hemorrhage. And in acute cases, this leads to fever, edema, coronitis. In some cases, to cyanosis of the tongue, so that gives us the bluetongue name, although that's actually quite rare, and death. There are 27 and counting different serotypes and many strains per serotype. There's not much cross-reactivity in terms of serology. Effectively, we have 27 different diseases called bluetongue, and they all have slightly different or very different characteristics. So it's a really different picture. This is some of the clinical signs of bluetongue. You can see the swelling in the top left image, of the sheep that's got a swollen tongue, swollen face. The erosion in the palate and soft palate and around the muzzle of those sheep, and unfortunately some dead sheep there. And then this is a kind of an unusual case that we had with Bluetongue 8 in Europe. We did actually see some cattle with clinical signs as well. So reddening of the eyes, reddening of the teats and around the nose, and a discharge from the nose. But most cattle will be absolutely asymptomatic. Just to demonstrate how dangerous this is to sheep, in 2006, Belgium lost 35% of its sheep stock to bluetongue. Belgium doesn't have that many sheep, but if this got into the UK flock, the sheep flock, it would cause huge amounts of damage. The transmission cycle of bluetongue is actually quite simple. So if a female bites a viremic ruminant host, it undergoes an incubation period called the extrinsic incubation period, whereby the virus travels from the gut through the tissues of the midge to the salivary glands. And that is a temperature-dependent process and lasts from 4 to 20 days. Obviously, the lower the temperature, the longer time that takes. And there is a limit to a thermal limit to the ability of bluetongue to replicate of about 12 to 15 degrees. The infected adult vector then can bite a susceptible ruminant host, and the host will then, after a latency period, become infectious to vectors after about 2 to 4 days, up to 7 days. So overall, the replication cycle is about 2 weeks. So from your first case, you might expect to see subsequent cases in the following 2 weeks, depending how warm it is. So as I said earlier, there's been significant changes in the epidemiology and distribution of Bluetongue virus. If I was sat here in 1990, this was the map we would have shown, and Bluetongue only occurred between the lines here between the 40 degrees north and 35 degrees south. It's a tropical and subtropical virus, and you just don't find it outside of this distribution. Since 1998, this has changed significantly. We've seen the emergence of exotic viruses in northern temperate regions, which I'll give an example of in Europe. We've seen an increase in global incidence of this virus, and an increase in the virus diversity in endemic zones. So we're getting more virus and different viruses even in endemic zones as well. So if we go back again to prior to 1998 and look at Europe, all Bluetongue virus outbreaks occurred to the south of this blue dashed line. And this is the northern distribution limit of the old world vector, Kilocortis amicula. We didn't see any transmission of Bluetongue outside of the range of amicula. And that was the status quo for, well, forever. Since 1998, we've seen incursion of Bluetongue into different regions within the southern Mediterranean. And when we went looking, we found new populations of Kilocortis amicula. I say new because they were new to us as researchers. No one spent that much time bothering to look for amicula in a lot of places before they had problems with Bluetongue. But molecular evidence shows that there's both recent in Spain and France and ancient establishment of Kilocortis amicula in Europe. And the BTV incursion pattern mirrors the genetic relationship between the Kilocortis amicula populations. So we have viruses coming from the east and coming from the west and coming from the south, and they kind of stick within the populations they arose from in genetically similar populations. So basically the insects have gone a certain direction and established along those in those countries, and then they've been followed by different Bluetongue viruses. In this period between 1998 and 2006, however, we found Bluetongue virus as Bluetongue virus outbreak in areas where there was no Kilocortis amicula. And what we think has happened is we've now got transmission in northern European midges. And amicula has kind of passed the baton by infecting animals at the edge of their range. They were then fed on by northern European midges, which could, at the middle or to the south of their range, and they were able to pick up the virus and then transmit it onwards within the northern European herds, or Central European herds in this case. Everything, I mean everything changed in northern Europe in 2006. Suddenly, Bluetongue 8 arrived in the lowlands of the Netherlands and Belgium and initiated an outbreak. At the time, everyone thought that this is very interesting, but perhaps kind of limited in its cost because this is a subtropical disease. There's no way it can overwinter because we have such a long period between our the end of our vector activity period in November and the beginning of the new activity period in April. So it wouldn't be able to bridge this gap and overwinter. We were proved very, very wrong. And in 2007, it overwintered incredibly well in several foci and initiated the largest and most economically damaging BTV outbreak in history. It was only by extensive vaccination programs in all the affected countries that we actually got a handle on this. And the UK was pretty much the only country not to have Bluetongue virus in 2008 and 2009 due to its really efficient vaccination program. So it's one case when vaccinations worked really, really well. I said it's the most economically damaging. You can see some of the costs. This is just from the Netherlands from 2006 and 2007, where we see kind of in the first year, £30 million in costs to the industry, exploding in the second year of the outbreak with a huge number of holdings infected. And although it's the sheep that are affected, it's cattle industry that has to be vaccinated that bears the brunt of the costs. Our more recent study has looked at a further breakdown of costs in Germany, and you can see we're talking about.
Chris, in the last slide, there was a large change in the profile. So because in 2006, control was responsible for the costs, and then 1 year after, economical losses due to the disease were. What changed from 1 year to the other?
So for a start, we had a huge number of cases. The actual number of cases went up vastly, and then the vaccination program started. So although the vaccination hadn't got in, the cost of that vaccine had to be added somewhere. So now we're talking about a big change in the approach because people realized they needed to do something about this virus because it was going to overwinter again, and it was going to continue to cause issues. So you can see that broken down a bit better in this slide, where in blue we now have the vaccination costs in 2008 and 2009 all the way up through to present day. So our direct costs are reduced after 2007. We're losing less animals, less productivity, but the costs are there due to the vaccination of huge numbers of animals to get this virus under control. So what do we know about the Bluetongue 8 in Northern Europe? It was a big surprise. It's not a result of the range expansion in Kilocortis and Mycula. This was being transmitted by Northern European midges. And the serotype was not on the periphery of the European Union. This came from nowhere. Somewhere sub-Saharan Africa is the closest match we have. It's probably not from wind-borne insects being blown into the Northern Europe, and probably not an animal import. So where on earth did this come from? And the answer is we don't know. Once the virus had gotten to Europe, it may be that a heat wave had actually helped the virus kickstart that transmission process with a little bit more efficiency. It was very hot in 2006, 2007 comparatively. Sorry, I'm speaking European comparatively hot. So it may be that that has initiated that outbreak. But as you can see, this year, last year now, in 2020, it's not just Bluetongue 8 that's come into the European arena. We have, we've run out of colors. That's how many viruses we have now circulating or assumed to be circulating in Europe. So these are all different viruses or different mixture of viruses where we got different serotypes co-circulating in different countries. And we have a real mixed picture, whereas 20, 25 years ago, we had 1 or 2 outbreaks in the preceding decade. In 2015, after all this fantastic vaccination, and we thought we were safe from Bluetongue 8, but BTV8 actually re-emerged in 2015. It'd been absent for 5 years, has had very few changes to the genome, but it appears that the pathology and the clinical disease that we see in the spread is very different to what we saw in 2006, 7, 8, 9. And a big question is where has it been, and why has it not spread? And I'll come back to that in a little while. So what do we do at Pirbright? Well, Pirbright was actually looking at Bluetongue virus and Kilocortis way before 1998, with John Borman and Philip Meller really championing this. Even though it's an exotic disease and no one thought it could possibly damage Europe, they pointed to the fact that it had caused small outbreaks in the Mediterranean islands, and it was worth studying for interest's sake, and how fortunate we were that they were doing that, 40, 50 years ago. At the Pirbright Institute, we have our high containment laboratories, so we can work with SAPO4, which is required for foot and mouth virus, and also BSL3 pathogens, so ACDP3 human pathogens. We can work both in the laboratory in high containment and also in animal containment, so we can actually study ruminants, hosts, and their infection with these viruses. We also have a dedicated arthropod infection suite, which does exactly what it says. We can use this suite to infect insects with these viruses up to ACDP3 and look at their transmission parameters in those insects. Now, most Kilocortis, I've said before, they're small and hard to work with, but most Kilocortis species cannot be colonized. It's a serial problem for students that someone tries to say, we'll get a student to try again, but no one's been able to do anything other than Kilocortis niveculosis from the UK and Kilocortis sonorensis from the USA, which are quite similar species, and they've been in colony for many years, so it started in the 1960s. Because of these facilities and the field work we're able to do, we've been able to look at all aspects of Kilocortis and Bluetongue virus behavior and ecology and interactions. From Kilocortis behavior in the field, their seasonality and abundance, down to laboratory experiments and looking at interactions with the virus, the vector, and the host as well. So this is where I started my journey on this, looking at Kilocortis dispersal. So a key part of the activity, the understanding of vector-borne disease spread is understanding the activity of the vector, and particularly how far they fly and why they fly between animals, how far they can go. With Bluetongue, we had historical outbreaks of Bluetongue virus on Mediterranean islands where there'd been no suspected animal imports. We suddenly have the arrival of a Bluetongue virus that's caused an outbreak, and the nearest source of virus would be on mainland Africa. And that suggested that long distance dispersal on the wind was possible. But one thing we don't know is how far can Kilocortis fly? Obviously, we're being an island nation ourselves. Is the UK at risk of that? How is this flight affected by season and meteorological conditions? And can we actually predict the windborne incursion or arrival of windborne BTV infected midges from the continent to the UK? So we did a bit of field work to understand the flight parameters of Kilocortis, and one of the first things I did was to fly this balloon. Many insects use the atmospheric boundary layer. This is a layer of basically laminar airflow above all the turbulence that's caused by buildings and trees and mountains and things. And a lot of insects use it for migration. So things like winged aphids, moths, plant hoppers, these migratory usually pest insects use this layer to migrate vast distances in a relatively short period of time. They do this because the temperature and wind speed is suitable for long distance insect flight. So you get temperature inversions, which is actually warmer in this boundary layer than it is close to the ground. So we use this 6-meter helium balloon. You can see me down there in the picture wearing my hard hat. And you release this up to around 250 to 300 meters into the boundary layer. I wouldn't recommend doing this as a sampling method for Kilocortis. We established their presence at a very low density. We caught 2, but we know they're there, and we know we caught them at that height because of the equipment we use. So The individuals we caught would have been transported tens of kilometers over the dusk period over which we collected them. So they are there, they are at low density. We're not seeing a mass migration of these insects, but we are seeing insects at altitude. So then we thought, well, what conditions allow them to fly? What conditions will cause them to disperse or allow them to disperse? Most of our collections of Kilocortis are light traps or animal traps, or something where we're drawing insects in. So we wanted to use a more passive collection method, and we used this truck trap, which we drove around a farm over a couple of summers. It collected an unbiased collection of Kilocortis flying at relatively low level. We ran this hourly over a main activity period and collected species level data for it, and showed that our Kilocortis in the UK are largely crepuscular, and there's limited variation in this around the season. So they were active a little bit earlier in the autumn than they were in the summer. This was great fun, and it gave us some really nice data. Now, I did my PhD at Rothamsted Research, which is a very similar institute to the Pirbright Institute, and but that looks at crops and crop pests. And knowing that I'd moved to Pirbright, they contacted me and said, well, we collect insects in our Rothamsted Insect Survey, which is a network of these 12-meter high suction traps. And they're looking for pest aphids to monitor the incursion of aphids that carry viral diseases of crops. And they collect Kilocortis as a bycatch, and were we interested? And of course, we jumped on the chance to look at these samples. So we had 2,700 odd samples from 2008, over 51,000 Kilocortis, because we're collecting passively just what's going over the top of that trap. These are Kilocortis that are actively engaged in dispersal. From this, we were able to get a descriptive and predictive Bayesian model of activity with season and meteorological conditions. And you can see the seasonality component of that equation there. So this gave us a really, really good detailed information of different species of UK Kilocortis, and as I said, predictive model. If we have a certain set of conditions of a particular season, we can understand roughly how many Kilocortis might be airborne at that time. So in 2007, the proximity of Bluetongue virus in France and the continent to the UK was an obvious threat to the British livestock industry. And unfortunately, in September 2007, we had our first case of Bluetongue virus. It's Debbie, the Highland cow, was on a rare breeds farm. It was diagnosed as BTV8 at the Pirbright Institute. And our environmental agency came to us and asked, was this a probable wind-borne incursion? Because we hadn't imported animals to this farm at all. And we can use kind of modeling of the winds that was present, and we can match actually an incursion event that probably carried infected midges from the continent to the UK over the correct time period, which is a really nice demonstration of a collaboration between ourselves and the Met Office, the UK Meteorological Met Office. And this is done by using the Met Office's numerical atmospheric dispersion modeling environment. And basically what this modeling environment does is release theoretical particles into a theoretical atmosphere that undergoes the same conditions as the atmosphere is going through in real time. And it's used for describing the particulate fallout for things like volcanic eruptions, for radiological escape events like Fukushima, and large fires, to understand where these particles are going to go in the atmosphere. And within this environment, we can actually model these particles as Kilocortis and give them behaviors such as takeoff time and fallout due to things like precipitation and a maximum takeoff wind velocity. These insects are very sensitive to wind speed and rain and temperature. And we can incorporate that into this model. And what that's given us is a predictive and descriptive tool that's actually used by UK policymakers to assess the risk of Bluetongue incursion into the UK in close to real time. So they ask the Met Office to run this model when they think there might be a risk of Bluetongue incursion into the UK, and we can identify those areas in the UK that might be at risk. This is an established part of the UK response to Kilocortis-borne virus threat. So that's dispersal over water, where the consequences of ceasing flying for an insect are fatal, but we don't see those big jumps of Kilocortis-borne diseases over or viruses over land. The spread of BTV is usually less than 5 kilometers a day. So obviously a different process is taking place over land. We wanted to understand this too. Most flights of these insects are going to be fairly local. They're going to be response to appetitive cues, so towards mates, towards hosts, and towards larval sites. The traditional way of studying this would be to look at using capture mark recapture, which has been used since time immemorial. But with Kilocortis, as you can see on that image on the right, this is problematic. They're very small. They get clogged up with fluorescent dusts and dyes, and we get a very low recapture rate. Less than 1% is typical. And it's very difficult to make any predictions based on a very few individuals, even if you can release hundreds of thousands of them. There's also the issue that their behavior might not be natural on release. And certainly when we collect insects at light, a lot of their natural behaviors are switched off. I mean, it's very difficult to get them to feed even on animals, for example. And what's the impact of meteorological conditions? This is not often studied, especially when you've got very few data points to look at. So we thought, how can we eliminate this capture step, which is probably the biggest problem for these sort of studies with Kilocortis. And by using immunomarking with a cheap food-derived protein and a highly sensitive ELISA.
Chris, maybe I lost something. So you mentioned the recapture, but there are what type of bait you use to trap to recapture the insects after release?
So nearly all our collections of Kilocortis are at light. So they have a light suction trap, which is a UV or black light like suction trap. It's very efficient, very effective for collecting Kilocortis. Some species have slightly different preferences. Some species need CO2, and some species prefer green light, but generally the gold standard is the UV black light trap.
Okay.
I had a pitch one for one earlier. I shall point it out afterwards. So yes, so if we could mark these insects in the field by spraying the habitat, we could actually avoid the capture step and cause minimal disruption to the typical behavior. So we developed this in the lab, and we sprayed Kilocortis larval habitat around a barn with 20% egg white solution. And then we collected Kilocortis in these CDC UV light traps in radial transects across the farm and linear transects to neighboring farms. And then we used high resolution weather data loggers. When we collected those insects, we tested each individual for the presence of egg white ovalbumin. We could use ovalbumin because there weren't any chickens on the farm, which is very useful, as it turns out. So we collected over 9,000 Kilocortis and tested them. And we had 600 positive individuals, which is really, really pleasing. There's a nice picture of a trap in that in the image there. So it's a little black light that's kind of then sucks into a collection sock and tube at the bottom. So of those 600 positive individuals, the majority, so 94% of them, were actually collected in the within the vicinity of the marked area. So most of these insects are not flying far before we collect them. Maybe they would have gone a bit further if we hadn't collected them, but most of them have stayed pretty much, which is what you'd expect, they've got everything they need within a short space of time on the farm, within a short distance, sorry. But 6% of those marked individuals were collected at distances greater than 1 kilometre from the source. And we observed upwind and downwind flight from our collections with a maximum distance of 3 kilometres from the source in 24 hours. So these insects are actually travelling quite large distances overland in a relatively short period of time. Now, once we've shown this, our next thought was, would this work for other small vectors like sandflies? And this is where our collaboration with Fernando and Rod Dillon and Luigi Seda at the University of Lancaster to try and get this, this technique working with sandflies. But that's a story for another day. So one of the things about the emergence of Bluetongue in Europe is it really has been elevated as a prime example of the impact of climate change on vector-borne diseases and what we might be expecting to happen to the distribution of vector-borne diseases across large areas of the world under climate change scenarios. This correlation has been made in Europe, where we've seen warming in the areas where Bluetongue we have had outbreaks of Bluetongue. But is there actually any evidence for changes in the European Kilocortis populations? It is very difficult to get long-term data sets that you need for this sort of analysis. And no one really looks at Kilocortis unless you have a problem with Bluetongue virus. So we were very lucky we could return to Rothamsted and their historic Rothamsted suction tract collections. So not only have they been going since the late 1960s, in fact, but they've also retained all these samples. So it's a fantastic collection, unmatched in the world for a lot of different species. And fortunately for us, Kilocortis, and the Kilocortis are actually in pretty good condition. You wouldn't want to do molecular analysis on them, but you can certainly identify them as a species group on the wing patterns. So we took Kilocortis from two sites where they're most abundant, in Preston in the north of England, and which is just near Lancaster actually, and Starcross down in the south. And from 2,800 samples, we pulled out 150,000 Kilocortis. And over this kind of 40-year period, what we saw was that at the Preston site, we had a much earlier appearance. There was half a day earlier per year in the analysis, and a later last appearance. So these insects were actually, for every year of the study, their activity season had increased by 1 day. We also saw an increase in the maximum and mean catch of around about 5% per year as well. So not only are they around more, there's more of them. And this is a case of repeating stuff is always interesting in science. But when we got to the Starcross site, we saw no significant changes. So why is this? And what does it correlate with? Well, the changes at Preston actually correlated with the climate change we've seen in the UK. So we've seen increased temperature and precipitation at the Preston site. We looked at a lot of other cofactors, so things like land use, how the amount of pasture and the host density, so the number of ruminant hosts that are available in the area, and therefore the habitat that's available to them. And those factors hadn't actually changed all that much, or if anything, it would be negatively impacting on Kilocortis populations. And so our theory is that the temperature change at Preston affected the population, which was actually able to increase in size because of the availability of precipitation and that moisture that they need in the environment. Whereas this was limiting at Starcross. So despite the fact they got the same temperature increase, didn't get the same precipitation. And so the population was limited at the same level. And the take-home message from this is that climate change impacts on vector-borne diseases will be determined by local factors, and sweeping statements of, saying we'll get an increase of vector-borne diseases across large areas like Europe, for example, is oversimplifying the situation. That said, if we look at the transmission season of Bluetongue virus in Preston, the number of disease cycles has actually increased, potential disease cycles, I should say. So we have 40 days longer period of disease transmission in Preston now than we would have had in 1974. So an additional disease cycle. And maybe that's why we are now seeing the increase in Bluetongue virus in Europe is that the overwintering period is much shorter, and so these viruses are actually able to stay establish and stay in Europe. Okay, let's be quick. So now moving on to Kilocortis and BTV and ruminant interactions. So I mentioned earlier that different Bluetongue viruses, the serotypes and strains have different phenotypes. So among the 27 different serotypes, loads and loads of different strains, each isolate can actually behave quite differently. And that has impacts on the emergence and spread. So how far these viruses spread in different regions. It can impact on their replication in the host, in the insect, and also therefore their transmission, and also can affect the clinical disease we see in the ruminants, their pathogenicity. And some traits are associated with specific genome segments. So Bluetongue virus is a double-stranded RNA virus with 10 genome segments. And these genome segments encode for the proteins that make up either the BTV structure or non-structural proteins that are involved in the entry, exit, and replication of this virus in host cells. And obviously, being an arbovirus, it has the two hosts to contend with. It has the insect host and the mammalian host. So sometimes we can get sort of co-infection of animals and indeed of cells with two different viruses. And because of this segmented genome structure, Bluetongue is actually able to exchange genome segments during co-infection. And this can happen in the cells of a ruminant host. So here we have a sheep that's been bitten by Kilocortis carrying two different viruses. Within the sheep, those viruses will replicate, and in some cells we may get co-infection. All those viruses and what we call the virus swarm will proliferate at different rates, and it's possible that a midge biting that animal may pick up a virus that actually behaves quite differently to the virus that from the parental strains. And the same can happen in the Kilocortis host, that a Kilocortis could feed on two different animals that have two different Bluetongue viruses, and that reassortment and the bottlenecking of those viruses can happen in the Kilocortis, and it then transmits something very different on to its subsequent ruminant host. The probabilities of that happening in Kilocortis is quite slim. So reassortment may affect the phenotype, including the pathogenicity and transmissibility of a virus. So this swapping of segments. We may see differences in the extent and severity of clinical signs, and we may see differences in the vector competence. So it's the proportion of individual Kilocortis that are actually able to transmit the virus. I should say, I think I forgot to say earlier that Bluetongue virus is typically very inefficient to get into the Kilocortis vector. So typically, we see less than 1% of the population that is fed on a viremic animal being able to then subsequently transmit that virus. That is compensated for by the fact you've got hundreds of thousands of insects feeding on these animals. So although the vector competence is very low, the transmission is possible just because it's a numbers game, because you have so many insects that are fed. Interestingly, the transmission from infected vector to the ruminant is very efficient. We'll see an example of that in a sec. So to look at this in the lab and in animals, we looked at the effects of reassortment on the phenotype of these viruses. And we took 3 parental strains of viruses that have been isolated from the field. So there's a BTV4, BTV1, and a BTV8. And then 2 reassortment strains that had so genetic backbone of one, but segments of another, or 2 other viruses from these 3 parental strains. So the similarity of the segments is really tight and very similar on a segment basis to the parental strains, but have mixed and matched. And hopefully you can see that by the table here. So we either got 2 or 3 reassortments strains represented. And this is our BTV Kilocortis ruminant infection model. We can actually use Kilocortis to infect ruminants, either sheep or cattle, in our isolation units. And this gives us a very natural infection system, because we know that inoculation by needle doesn't replicate the infection we see in the field, and it does change the pathology and clinical score that we see. So for each sheep, we had 4 different sheep for each BTV strains. We have 5 strains of Bluetongue, 4 sheep per strain in 4 replicas. It's quite a large experiment. And we inoculated Kilocortis with a standardized dose of Bluetongue virus and then incubated them. So we've bypassed this issue with the low vector competence of some of these strains. We'd have to feed thousands of them to get enough to survive to actually infect a sheep. But we can inoculate hundreds of them, get some to survive through incubation, so we know they're infectious, and then feed them on a sheep, monitor that sheep, and at peak viremia, shown by the PCR of the virus in the sheep's blood, we can then look at onwards transmission from the animal, feeding directly on the sheep, and also taking blood from that sheep and doing artificial infections in a hematec or a blood reservoir system at a later date. And what we found was that our infection success with inoculated sonorensis, Kilocortis sonorensis, was very good. All of the Kilocortis we infected that actually fed were infected with Bluetongue virus. And all the sheep that we exposed to those infected midges came down with Bluetongue virus and showed viremia. Now, the survival rate and the feeding rate of those surviving inoculated midges varied wildly, from cases where we had just 1 or 2 individuals being able to feed up to 70% of those surviving insects being able to feed. And that is part of the issue of inoculating. You're stabbing a very small insect with a comparatively large needle to get virus into them. So it is a very delicate process, and we do lose quite a number of insects by that way. You get better over time, essentially. But what's interesting is that when we look at the viremia of these animals, so this is the CT value, the PCR results for each animal, and we don't see an effect of BTV strain on the timing of the peak. So they all seem to come up with their viremia roughly at the same time. But we do see an effect on the level of viremia, largely because some of these viruses were particularly nasty. You can see BTV1, we have a very high viremia, and actually none of those, all of those animals had to be euthanized because they were too sick to continue with the experiment. So there is some effects of strain. Interestingly, there's not a lot of effect on the number of Kilocortis that bite. So even with very low numbers, so 1, 2, or 3 infected midges feeding, we still get an efficient infection of that animal. So it only takes 1 infected Kilocortis to bite. And we don't see a big difference if there's 50, 80 midges that are actually feeding. But what we do see is a relationship between the number of midges feeding and the level of the peak of viremia in the animal. So the amount of virus that's able to develop. And maybe that's because the virus dose is lower and the host's immune system is able to get on top of it a bit quicker. Maybe there's lots more to understand about that process. In terms of the sheep themselves, So clinical disease in infected sheep varied with the strain quite considerably, with some of the parental strains showing very high severity of disease. BTV1 and BTV8 were quite severe, BTV8 slightly less so. And BTV4 parental strain had very low severity of disease with very mild signs. And the reassortant strains were actually intermediate between these. And it shows that the causation of pathogenicity, the causation of these clinical signs is multifactorial in terms of the genome of these viruses, and is not dependent on their serotype. When we look at those insects that have been fed on those sheep or on their blood via membrane, I can see I've highlighted here how low the vector competence is for BTV8, where we didn't get any individuals positive, or just 4 out of 1,000 individuals positive on the sheep. So 0% Kilocortis. This is the virus that spread right across Northern Europe, and it has a very low vector competence. Interestingly, we have a very high vector competence, and this was really quite surprising. We hadn't seen a vector competence rate this high in a field or artificial setting. So getting up to sort of 50, 60% competence levels in the reassortant strains. This has opened up a whole new amount of work that we can do with this strain, because now we have a competence level we can work with. But susceptibility of infection of Kilocortis and their vector competence for these viruses varies significantly with the BTV strain, and can change rapidly via natural reassortment. So we can see that if you've got more individuals capable of transmitting this virus, this is going to change the transmission and potentially the spread of that virus. Having said that, both these reassortant strains were restricted in range to Morocco and didn't explode beyond it. Why is that? Who knows? Again, lots more work required to understand that process. If we have a look at the disseminated infection, so the previous slide was just positive individuals, but we can actually use the CT value to understand whether these individuals have actually a fully disseminated infection. The virus was actually in the head, in the salivary glands, and ready to transmit. And you can see 2 individuals there in BTV8 that I've highlighted. Those 2 individuals had a pretty low CT value with a probable, from our previous work, shown it's a probable fully disseminated infection, and they would have been able to transmit the virus. You can see all the others in the other serotypes below that line are far higher, whichever threshold you use, we have far higher infection rates and probable dissemination rates in images in the other serotypes, and particularly the BTV4 reassortant strains. Obviously, this is with our pet Kilocortis sonorensis from our colony, which has been in colony for a long time. And we did have to check that this is actually a true representation of what's going on in the field. And by and large, although we swapped hosts here, now we're talking about cattle, we repeated the experiment with BTV1 and BTV4, sorry, reassortant strain. And we see a very similar relationship between the BTV1 and BTV4 with field collected Kilocortis. The issue with this is that you have to collect Kilocortis at specific time points in your animal experiment when the animals are at peak viremia, and you are practically guaranteed it'll be raining, it'll be stormy. The delights of field work combined with lab work are a complete curse. So although we get a relatively few insects, and that's probably associated with the, you can see that from the data there, the percentages, there's quite a lot of variation around that. And the feeding rates from these insects, as I said earlier, is actually quite low. But broadly speaking, we see similar susceptibility in field populations as we do in our Kilocortis sonorensis. So moving on back to what happened with BTV8 when it re-emerged in 2015. So just to reiterate, it had been absent for 5 years. The vaccination program was very effective. We had not seen a single case of BTV8 in Northern Europe for 5 years. Then 2015, the strain re-emerged, it was very similar, with 11 amino acid differences from the very first strains of the initial outbreak. So we would have expected more amino acid changes in that genome over the period of time, especially over those 5 years when it disappeared. So we put this back into our animal model. This time we did a needle inoculation and then monitored the two groups of sheep given either the 2015 strain or the 2007 strain. We demonstrated that these animals displayed reduced clinical signs, so they're less sick than those previously. And they had a reduced viremia, so the level of virus in their bodies was reduced. And this probably in turn caused a reduced vector competence. So there's less midges that are able to transmit this virus. And this might be why this virus hasn't displayed the same phenotype and the same behavior as it did in 2006, 7, 8, when it spread right across Northern Europe. It's still going. We still get reports of cases coming up in France and in the countries bordering France, so Switzerland and Germany in particular, but it hasn't shown this kind of rapid explosion that we saw in the first outbreak. So those very few changes are perhaps enough to confer a different phenotype on this virus. And certainly it means that we're less worried about this virus than we were initially when it re-emerged in 2015. Okay, so in summary, the dispersal of Kilocortis vectors can be predicted over long, greater than 30 kilometers, and short distances. And we can show that with the impact of meteorological conditions on their dispersal as well. The impact of climate change on the seasonal activity of Kilocortis and the BTV transmission season will be moderated by local factors. So, broad sweeping statements of the impact of climate change may not demonstrate what's actually going to happen on the ground. There's going to be some places where they see large impacts of climate change, others where it may not actually be impacted at all because of these cofactors that are really important at the local level. We've seen that small changes in the Bluetongue genome and reassortment between field strains are likely to affect virus spread and clinical outcome. And as a result, we can't use serotype as an indicator of the phenotype of that virus. So just because we've got BTV8 or BTV4 doesn't mean we can expect the virus to behave as it has in the past. And we've seen that the genes and proteins responsible for variants in phenotype cause quite a mixed picture. Even from the data we haven't been able to pin down exactly what the differences are, even though we have full sequences for those viruses. So we need to go back to those strains and actually find out what's responsible for those phenotypes that we see. Broadly speaking, we know which protein segments are responsible for which part of the infection of cells, but which bit is responsible for the infection in mammalian rather than Kilocortis cells, for instance, is still under investigation. So I thought I'd just give an overview of what's happening next in our group. So there's a number of projects that have started or are about to start, that are ongoing at the moment with Kilocortis. One of the most important questions we have is how does Kilocortis-borne viruses like Bluetongue, like Schmallenberg, overwinter in the UK and Northern Europe when we have this period of absence of the vector? Obviously, the animals are still there. So how long is a BTV infected animal infectious to Kilocortis? We've not been able to explore this before because the low vector competence of a lot of these strains. We're struggling to get any insects infected at peak viremia with a lot of strains, but now we have the opportunity to use high vector competence strains where we're expecting sort of 50% of those insects infected to be able to transmit the virus. And so we can look at how that wanes over time. And this is important too for cases where we've had the import of animals that are tested by the reference laboratories. So they come with it into the UK and then they're tested. They're supposed to be tested prior to coming to the UK as well. We test them, and if they have Bluetongue, they raise alarms and are destroyed. But what risk do those insects pose when they have either old infections or just about detectable infections. That's something we need to know. Something else we're beginning to study is the influence of endosymbionts like Wolbachia and on Kilocortis vector competence. So what is the role they play, if any, and how does that interact with the different species that we have and different viruses that we have in circulation? So you'll probably be familiar with the impact of Wolbachia in Aedes, and dengue virus is a fantastic tool that people have been able to use for dengue mitigation by reducing the population of competent individuals in the Aedes population. So understanding what's happening in Kilocortis would be really interesting in that as well. I don't think we're going to be able to get to the point where we have an effective field tool just because of the issue with colonies, but it's certainly interesting and thing to look at. Something completely different, perhaps, is the study that we've literally just started, where we're looking at to see if infection with Bluetongue virus and other viruses changes the volatile organic compound profile, so the smell, essentially the body odor of an animal in a specific way. We've got some good evidence from malaria, and you might be familiar with the fact that dogs are being used to sniff out COVID in people at a number of airports in Europe. So we know that there is a detectable smell of some viruses, and some diseases, the animals you can smell yourself, and we've got a really insensitive nose, so as humans. So by using some sophisticated collection methods, we're going to look at see if the volatile profile of animals is actually impacted by infection. And for vector-borne diseases, this is really interesting. So if it does prove to be the case that these animals have a changed profile, does that mean anything are they bioactive chemicals for the vectors that are olfactory in their responses to animals are completely olfactory? Does that actually have an effect on the preferences, the biting rates, the host choice of those insects? So really exciting to start that one. And then finally, I thought I'd show you some nice pictures that are hot off the press. These very recent pictures that a colleague of mine, Mark Gamera at Pirbright, and I have generated to visualize the infection and dissemination of BTV in Kilocortis tissues. People have done this in the past by using cross sections and amino labeling, and we've taken the antibody labeling a bit further and using confocal microscopy and excise tissues. And this is the salivary gland of a Kilocortis sonorensis 8 days post-infection. And you can see DAPI staying as the blue of the nucleus, and the tubulin in red, that giving you the structure of the sort of bag-like salivary gland. And then in green is the virus structural proteins. So this is actually Bluetongue virus in inclusion bodies in those cells, ready to be kind of transmitted, in this case, from the salivary glands. And not only in the salivary glands, we've also been looking at the midgut, you can see on this the top slide. And then we know through previous studies that Bluetongue virus is not transmitted vertically in the sonorensis host. And it's nice to show that we don't actually see any Bluetongue virus. This is the same individual. We don't actually see any virus in the ovary or in the ova that are ready to be laid. So something exciting to end with, very preliminary stuff, but some excellent images I'd like to share with you. Finally, there's an awful lot of people I'd like to thank, both at Pirbright, past and present members of the entomology group, Orbivirus group, and reference laboratories. The Met Office, long-standing collaboration with them, and also Rothamsted Research, and of course my funders. Thank you very much for your attention. I'll happily take any questions.
Well, thank you, Chris. I think in normal conditions, you will have clapping and congratulations. So I have loads of questions to you. So, but let's see if the audience have any questions before. Maybe in the chat. I have to look in the chat. Okay. Well, I will start for warming up and see what happens. Talking about, I was curious. There are many studies showing that in Brazil the mosquitoes travel by bus, in the cars, maybe in ships and planes. Do culicoides do the same? Do you have any evidence for that?
No direct evidence, but lots of supposition is the answer to that. So one of the when Bluetongue 8 arrived in Northern Europe, that no one understood where it come from. No one had any data of anything similar on the periphery of Europe. So the only places where Bluetongue 8 had been found previously was sub-Saharan Africa. And one of the biggest imports from sub-Saharan Africa to that part of Europe is cut flowers. So for domestic use, so for flower arrangements and people to have in their homes. They are cut at night when Kilocortis be active, and they are packed under lights. So you can see you'd be attracting lots of Kilocortis, and then they are chilled, which would increase the survival potential of those Kilocortis. And then they're unpacked at big flower markets in Europe. It's a very nice just so story. I do know some people have been out to Kenya and places where they do these packing, and it's plausible, but actually finding physical proof of Kilocortis, I mean, you're looking for a very small needle in a very large haystack, or flower stack in this case. So yes, probably. There is transport of Kilocortis along with hosts, with on aircraft and container shipping. I mean, part of the reason that we have all these viruses going around the world and COVID is because we're so interconnected, and so global trade will facilitate this, and there's no doubt of that. But actually getting hard evidence of Kilocortis transmission, Kilocortis transport like this is very difficult to do.
Well, that was done in Brazil using genetic markers. So studying population genetics and showing gene flow among populations. So maybe, don't know.
Yeah, we have looked at genetic markers like that using molecular markers and sequencing. One of the things it's shown is that the connectivity between the populations in Europe is really high. So as we showed, sort of 6% of those images we marked were traveling quite large distances, kind of almost between farms. That's happening on quite a large scale, and perhaps our experiment actually underrepresented the amount of movement that we see, because these populations are really closely related, much more so than you would think from the average dispersal distance of these insects. So yeah, it's very difficult to then look at kind of how far things have moved. Maybe we're not looking on a big enough scale. Certainly between the continent and the UK, I don't think that 30 miles is enough. I don't think the English Channel that separates the UK from the continent is a big enough barrier to stop significant genetic mixing of those populations.
We have questions from Reggie. Reginaldo Brazil is watching us, and he wrote that he has 2 questions. Please, Reggie, can you step up and make your questions? Your microphone is off. I can't hear you. Somebody is hearing Reggie, or?
No, not here. Reginaldo?
Reginaldo, a gente não te
Hello?
ouve. Deve ter algum problema no teu microfone, porque ele aparece para. Sorry, Chris, for the Portuguese. It's just solving. I can see you, Reggie, but I can't hear you. Are you using a phone or no? Now it's off, so you have to turn it on. There is a small button in the left. Can you speak something? I can't hear you. No, no,
No,
it's not working. Maybe if you take off the earphones, maybe with directly in the computer, it might work.
Can you hear me, please?
Yeah.
It's
Now it's
better
good.
now.
Now we are good.
I'm sorry, Chris, about this, but
That's all right.
thank you very much for your talk. A very nice talk.
Thank you.
I'm a sandfly man. I'm not Kilocortis man, so maybe my question is really rubbish. So, but anyway, first, Chris, just a question: do you have evidence of diapause in Kilocortis?
Sorry, could you repeat that?
Diapause.
Diapause. Interesting question. So we don't think that the adults diapause per se. We do see a very sharp reduction in activity in the winter in Northern Europe. But we do see some activity on warm days. So we don't think it's a diapause. We think that the overwintering stage is the larval form,
Okay.
or the egg form. But we can find larvae through the winter, and we can't find Kilocortis adults. Whether we're looking in the right places. One of the big things we've been looking at recently is overwintering in animal housing. So you've brought the animals in because it's cold outside, and you generate a warm atmosphere, a warm microclimate for them to overwinter in. And maybe they bring in Kilocortis with them as well. It'd be perfectly reasonable to think that. And certainly we do see some adults active, whether they've emerged and are active, or whether they are long-lived individuals that have kind of, as you say, diapause and then come out of diapause to go back in again. We're not sure. One of my colleagues is looking at age grading to find out how old some of these insects are and how old they can be, to understand the endurance of these insects. It's one of those questions where we thought we knew everything because we thought we knew that they all overwintered as larvae and there was no adult activity. And whether that's changing or whether there's always been activity just at a low level, we're not sure about. There's only been a very few cases of transmission of bluetongue over the winter period in Northern Europe. And they have been in really large internalized herds of cattle where, the cattle don't see daylight, so they are completely enclosed in a very controlled environment. That's the only places we've seen transmission of bluetongue.
Okay. Chris, thank you. Another question was about the nose. I know that people is being quite good results in leishmaniasis with the dogs. I think it would be really good to see if this virus in sheep can give you some kind of odor, and you can select them. That would be really fantastic.
Yeah, it'd be really exciting. So it's a brand new project that we've only just started. We actually employed the new postdoc on it yesterday. So so we will be infecting animals with Bluetongue virus. what's really nice is that we can infect sheep and they will show clinical disease, and we can get an odor profile for them, but we can also infect cattle knowing that we're very unlikely to see clinical disease, but they are infected with Bluetongue. So we'll know whether we get a profile change due to the infection with Bluetongue, or whether it's because of the disease that we're seeing. So the clinical signs and things like fever, you can imagine, will elevate the smell of the host because it's hot, it's sweating more.
Yeah.
But if we see changes before that, or in the animals that don't actually show clinical signs, that will be really interesting.
Yeah.
As you say, dogs can do it with leishmaniasis, and with malaria and with COVID. So we've got really good hopes that this will actually work. And then the onwards work for looking at the impact on Kilocortis is really exciting. That's what really gets me.
Okay. Okay, Chris, thank you. Success in your work, okay?
Thank you.
Thank you. Thank you, Fernando.
Reggie.
I don't know how I reduce my screen here. Fernando, sorry.
It's fine. No, don't bother about that. Now we are just chatting. Following Reggie's question, do you have any evidence that the midge's behavior changes with the infection of the vertebrate host? Do they prefer infected hosts or something like that?
No, we don't at the moment, and studying it would be quite difficult. Yeah, because we'd have to have an infected animal at the right stage, and something we hope to do in the future. The only evidence I have is purely anecdotal, from a farmer who said that it was only his black cows that got infected rather than his black and white cows. So there's a host preference there, that the other black cows got bitten more, or he was just, yeah, pulling my leg. I'm not sure. But considering most of his cows were a fair shade of, had black and white spots, I don't know whether he was or not.
Okay.
But yeah, whether there's a difference between the two, I mean, I hope to be able to explore that in the lab. Using the odor profiles with wide tube bioassays. I don't know if you're familiar with the choice chamber, so you can pass different odors through once you collected them, and then going into a kind of semi-field experiment looking at biting rates on animals would be really quite interesting.
You know that if this odor profile works, it will kill the job of a lot of molecular biology, you know. No?
I'd be quite happy if we could walk around with a dog and it barks when we got Bluetongue. That'd be so much easier. And,
okay. Let's. I don't know if somebody has just one more quick question. I know that it's late in the UK. It's
don't worry.
But I was curious about this thing in the atmosphere. How I understand that the Kilocortis go into this high atmosphere just passively. It's just they are just blown and carried with the wind. Is that right?
Yes. So the term aerial plankton that some people use implies that they don't have a choice, and they're not. It's not a behavior. They're just caught. Which is not true of plankton. It's not true of aerial plankton either. What we think is going on is when you get a large abundance of Kilocortis, they're highly active, and then you can get updrafts which would carry them to a higher level. Whether they're intending to fly miles, I doubt it. But over there.
Yeah.
So whether they're intending to disperse over long distances and that is inducing this behavior, and they actually facilitated by the wind, or whether it is a case of they're there by accident, we don't know. We can't tell the motivation.
In these conditions, in these airborne conditions, how long the adults are viable? Do you have any idea? So because one of these ice creams can take 1 week or 2 weeks.
Exactly.
Does anybody do you have these measurements?
So the biggest threat to the individual midge in an airstream is desiccation. So if the humidity is high enough, they've got sufficient fat stores and energy stores, they can keep going for quite some time. I've had them flying on a hare, so I attached some midges to a hare and had our colony insects, which haven't had to fly for hundreds of generations, but they were flying for 8 hours. If you just kind of keep touching their tarsi and allowing them to fly, just keep stimulating them to fly, and they will fly for 8 hours, which is quite impressive. But I think it's
but in this
carbohydrate.
But in this case, they are not actually flying; they are just being suspended. So
yes,
so
yes, you could argue that in an air packet they are also suspended. So yeah, I don't know how you would ever find that out without some impedance from, I mean, if you're looking at a large moth, you can use a flight mill and work out how far they can fly for on a flight mill, but with the inertia, it's just too big for Kilocortis, they're too small. So yeah, I think we have to go to the desiccation bit and the energy use of how long can they survive without a sugar fuel, and that's typically 24 to 48 hours. They're happy. So once they've used up their initial fat reserves, they are quite sensitive to that.
But this depends on the temperature.
Yes, very much so.
So you must have an idea of how long they can survive in that temperature.
Yeah, so at cold temperatures, they can survive a very long time. 6 months even at if they're held very nicely at ideal temperatures with carbohydrate source and things like that. At plus 6 degrees or so. So yeah, temperature is probably not limiting unless it gets too cold and they stop flying because they're unable to fly because it is too cold. And a threshold for threshold temperature for flight is around about 4 degrees. So we've done some work recently on looking at the temperature, the threshold temperature for flight, and whether these insects are actually active, or we collect them in catches in insect collections at light when the temperature is above 5 to 13 degrees. But the insects are actually able to survive much lower temperatures than that, down to just above freezing, and even freezing for a very short time. So yeah, one of the things we can't do is work on chill tables. So the Northern European midges just get up and walk away on a chill table, whereas your normal kind of Aedes or tropical mosquito will be out for the count and you can do whatever you like to it. Northern European Kilocortis will just shrug it off. So so yeah, we can't use chill tables for that. Yeah, the whole kind of process of getting up into the atmosphere and sustaining flight is really difficult to actually interrogate experimentally.
Yeah, I was wondering if you have some life parameters estimation in these conditions that so you can have some estimate that they can be viable for weeks, for example, when they are just flying over and
Yeah, sure.
I got this. So sometimes there are weird things going on and we have no idea of
yeah,
how much kilometers high they are flying?
So how high? So the boundary layer varies in height according to topography. So over the Alps, for example, over the mountains, it will be very high if it's there at all because you've got so much turbulence And so that will probably form a barrier. And typically in the late spring, early summer, the boundary layer in the UK is somewhere between 150 meters and a kilometer and a half. So it's actually quite high. And what you get is an actual stratification of insects in that airstream. So Rothamsted research have been looking at this for many years using radar. They can't pick up Kilocortis because they're too small, but they can pick up insects greater than 10 millimeters in length. And they have been able to see clouds of insects passing over, and I able to identify them by their wing beat frequency and size, and know their direction and things like that. Very clever stuff. But so you get this stratification according to temperature, and that's quite tight as well. So all the insects are using that layer, but that layer, the height of that layer varies with meteorological conditions on the ground. So, excuse me, the best example is when you get a temperature inversion where the ground cools and the warm air gets lifted. And so you get a warm pocket of air that you can actually measure using meteorological balloons. And that is where those insects will be flying. So in that case, it doesn't matter how high they are, as long as they're in that air that's the correct temperature for them to fly. And it can be quite warm, I'm talking sort of 20 degrees above a temperature of maybe 5 degrees on the ground. It's going to be that stratified as well, because it's not mixing as the air at the ground is.
Okay, great.
Very interesting.
Somebody wants to make any questions or?
No,
Okay, Logulo, do you want to
Okay,
make a?
I think it's okay.
Okay, so Chris, I have loads of questions here, but I will not exaggerate. Sorry, I will make these questions to you later. I would like to thank you very much for your presentation. Thank you for the audience, and I hope to see you soon, alive and well. Okay.
Thank you very much for the invitation to speak. It's been a pleasure to speak to you today, and stay safe, everyone. And I hope to see you in the future soon. Thank you very
Okay.
much.
Okay. Thank you,
Thank you,
Chris.
Chris.
Thank you.
Bye-bye.
Thank you a lot.
Bye-bye, people.